So here we are. Let me get the files up for you. So I'm in the HIV workshop 2022, and then I'm going to data science. And then there's should be a new directory here if you pulled successfully called examples. Click on that. And I prepared 2 examples for you. So basically today we're not going to learn anything. Yes, stuff. You might learn some new things because I maybe have a straight command here or there. But mostly it's going to be to reinforce what you've already learned. So we're going to pull in some data files and we're going to clean them up and we're going to do some visualization and stuff like that. And if we have time, we'll start a really frustrating project where you get an Excel file with multiple headers and you've got to figure out how to parse it so that R can actually make sense out of it. It's one thing that I often like to show to, especially to biological collaborators, because often they give us Excel file with, multiple header lines and this is color coded and all that stuff, and we go like, I can do that. I can do something with that, but it will hurt me and take many hours of my time. So, it's also good to know how to do these things. Okay. Oh, and also, sorry, this. Next Friday, I think I didn't advertise this really well, but next Thursday, you have a choice. You can come in and do some more examples and exercises I give you, or you can bring your own data. If you have data files that you've been trying to work with in R and you're stuck on something or you need help or you have questions, bring it on. It's exciting and terrifying for me because I just remember when you show it to me that I haven't seen your data ever before. But it usually goes very well, and it's actually one of the really fun parts of this workshop for me at least, and I think it's rewarding for the people who actually do bring their own stuff. Just a show of hands, does anybody have anything in mind that they might bring? So a few, good, all right, great. All right. So the first example here is it's practical, and it's really just it's an HIV kind of demographic stuff, like how transmission, deaths, and such over different demographic groups. And the input file is kind of messy, and we're gonna have to clean it up. So so the file here is a practical example of data lending. That's what we call it when we get data files and then have to manipulate them in multiple ways to get them into a tidy format so that we can visualize and analyze them. Okay. If I start talking too fast, or go, say, hey, Janice, because I forget. And it's perfectly okay to ask me to repeat things. I have severe ADD, and so I have to ask people to repeat things. I'm sorry, I wasn't listening to you. Could you? So if you have that, or you have, not if you have that, but if you need me to repeat anything, I'll say it 100 times. Okay, so in this notebook, step by step, one cell at a time. So the first one, so I'm just going to click. I don't know if anybody, if the other instructors have done this, but use this little run button here, the green thing that runs the current chunk. So the first one is a tidyverse stuff. Yeah.
Oh, can you just make the text a little bigger?
Oh, like this text?
Yeah.
Is there there's a setting for that?
I think if you go view.
View, right.
Or maybe not. Nope.
See, I think there is some way to do it
I usually do.
I know I've had
Oh, I think if you do and Control Shift plus.
You think that will just
that'll do it.
Does that help?
Yeah, thank you.
Better. We'll move this over so you can see a little bit more of the code. All right. So that was just loading up tidyverse. I don't know what those red things mean, but I'm going to ignore them. I don't think they're important. Okay. So our objective here is we want to visualize the change in deaths of people with HIV over time for transmission in terms of transmission risk factors. So first we're going to get the data and try to understand it. This file is on your because when you pulled, it's in the repository, so it's under the files directory. And the first thing I'm going to do, have you seen Read Excel before, by the way? Okay, so this is another, I think I should, let me go back and show you. This is a, I loaded Tidyverse, but in addition to Tidyverse, I loaded this other library called Read Excel. It's a newer package that has a lot of very useful functions for manipulating Excel files, like, you know, skipping headers and reading sheets and stuff like that. So if you ever have, like, say, an Excel file with multiple sheets, you can use this library to get them into separate data frames. And so I highly recommend that. I have a student who was like, oh, I have to cut down. No, we don't cut and paste ever. Okay, so let's read it in and store it in this data frame we're going to call DF. Another example of me being a poor lead by example person, calling a data frame DF. We should be calling it something much more meaningful than that, but I don't like to type a lot. So. Okay, and just like the dimensions, so then just sort of like, well, what is this thing I read in? It's got 215 rows and 4 columns. And we just do a little bit of poke around, seeing what's in there. If I just look at the first 2 lines, it's not terribly helpful. I see that I've got 4 columns. It's year, category, group, and count. At least I see that one category is age of death, and the group looks like it's age groupings, right? So that makes sense.
Sorry,
Yep.
sorry.
No, it's perfectly okay.
There's an error that came up, but I forgot to run the first thing.
Yeah, no problem. No, I just wanted to see if you had a question. That's why I stopped. I don't mind people talking at all. Okay, and then if I look at the end of the file, okay, so we've got a different category, transmission category, female adult, transmission category, child. So what's really in this? Well, I could try summary. Gives me a summary of a data frame, but this gives me a summary of essentially of you get for numerics, you'll get like the distribution properties for the quartile, and for the characters, it'll just tell you, things like the class and the length, so nothing terribly useful there. There's another command called str for structure. We'll show you the structure of an object in R. It's actually very useful when you're looking at lists, because people can have very, very complicated list structures. And so you can get an idea of what's in them by doing this. Oh, thanks. Sorry, I skipped a cell. Don't want to be here. So at least this tells me, kind of, and so it breaks it down. Remember, everything essentially in R is a list, right? The data frames are lists. There's lists that have special properties, like every element has the same length, for example. So we know that it's a tibble. The columns are year, category, group, and count. It's giving us some idea of what, but it only, it's kind of just listing the first one. So that gives us much more information than we got from the head count. But today I want to learn even more about what is this category variable. So we have this really great command called distinct. And what it does is it shows you what are the distinct values for a given character, or any work with any data type, but if you have something that's like kind of numeric or counts, it's not going to give you anything meaningful. Usually you want something that's some kind of ordinal variable. So if I do that, get a little bit more information. So I've got categories. These are the categories I've got: age of death, current gender, race, ethnicity, and then 3 transmission categories: one for adult males, one for adult females, and one for children who are basically less than 12 years old at the end of the year. So then the count, right, is going to be the count for something for one of these categories. And within that, it's going to be, there are groups within categories, right? So age has several different categories. And then within that is going to be the count of people who died in that distinct category. And group. All right. So I just wanted to, like, I made a note to myself. So it said, look at current gender to see what the groups are. So this would be basically the same thing where I just say DF and then distinct and oh, sorry, no, filter first, sorry. I want to look at current gender, so I'm going to filter category. And so if I just filter out the ones, oh, no, I'm done. If I just filter out the rows that have current gender, I can see what the group looks like in a category. So I see male, female, transgender, male to male. So that's what my groups are going to be within the current gender. To give you a little bit better idea what's in here. It's another way to look at this and get a category is looking at distinct category and group. So you can ask for the distinct values for category and group, and that will give you all the categories and the groups within them. So this is kind of annoying because it doesn't quite fit. Yeah, there we go. Okay. So here we see category age of death, right? So I've got a bunch of those, but then it gives me the distinct categories within that. And then if I go to the next rows, down here at the bottom, current gender, it shows me those groups. And race, ethnicity. So you can see, so distinct will work. You can nest distinct statements. All right. So let's start cleaning up a little bit. First thing I'm going to do is pull out the transmission category. So I want to look at transmission categories. So I'm going to pull out each of the different transmission categories. There are 3 of them. One is male, one's female, and the other is child. So here I'm just filtering out by category the full name, transmission category, male, adult, or adolescent. If I'm really lazy and I don't want to type all of this stuff out, what I could do is a string detect. That's often what I'll do. If I'm feeling very secure about what's in the data, right? I might just do, because I know that the child category doesn't have male or female in it. So so I might do something like, instead of what's written here, I would do string detect category. So that's the column I want to look in, and the string I want to look for is a male. Or type it correctly, male, and we'll do the same thing. Yeah, so that will do that, and then the next statement just selects. So I'm using the select statement to pull out the year, which I want. I don't want category anymore because I've only got one value of category, so we've done that. So that's minus, that's taking that out. And then I'm renaming the column group to be what the transmission category is. And the reason I'm doing this is because I want that in there because I'm going to pull a bunch of data frames all together in the end. So I'm going to pull the males, females, and the children all together. Do that and save it in mail. Just to show you what this first part did, I'll just execute this. So that's pulling out all, so without having to type all of the whole transmission, I just want mail in there. I use string detect. Right. And let's just look at what the now looks like now. And that's what it looks like. So we've got years, and then this column that says transmission category, male, adult, or adolescent. That's the group. Okay, so within that transmission category, this is what the group. All right, do the same thing for females. It's the exact same code, except that we just pulled out a different transmission category. And then look at what that looks like. So very similar. So we've got year, we've got this new column with this new name and the count. Same thing with children, doing exactly the same thing, just a different category. So I also want to note, so, and a lot of times in our notebooks, you'll see that when we want to look at a data frame, we'll have this pipe to the head function, right? And I think you know, probably you're told, the head function shows you the first so many lines of a data frame. I don't remember what the, I think it defaults to 8, but you can put in whatever number you want and it will display that for you. It's really important, not for what I'm displaying here, because when you run the chunks in RStudio, it only shows you so many, and then you click an X arrow to get to the next few rows. But if you knit the notebook and you have something like that in there, it will print the entire data frame. If you don't put like something like head in there. So if you want to knit this notebook and you didn't have the head statement in there, and I had a data frame of, say 250 lines, I'm going to get, so that's just for you to kind of make note of that. If you do that, it could be a little bit. All right, there's the children. Okay. Now we're going to get this so that now we're going to change this. We actually want to make this a wider data frame, the child data frame, because like I said, we're going to combine these guys. What I want to do is I want to have the categories to go to different columns. Okay, so I'm going to make this a lot wider. So I'm going to have for each transmission category within the child data frame, I'm going to give the count values for that. Does that make sense? Well, hopefully it makes sense. Okay, so again, pivot wider. Remember, names from. So I've got this transmission category. Look here. So transmission category. In this case, actually, shouldn't have done the child first. There's only one, right? The only transmission category for the children is perinatal. So I'm not really doing a whole lot with this first one, but you'll see it in the male and female. It's a little bit more, it makes a little bit more sense. Where did I go? Sorry. There we go. Okay. So show you what we did. So here we only created one column. It's called perinatal, and then we took what the we took the count and put that as the value in that. Right, that's what pivot wider does. And then we created a new column called category that gives us that just has the value child in it, and that we do with the mutate command. So it was two things combined, right? First, we made this thing wider, and we had these two columns, and then we added another column that has the category for child. And now I'll do the same for female, and you'll see a little bit more what I mean. So this is what pivot wider did. So it took each category and created a column from that column, from the names in that column, right? So it took the names, took a column of names, turned it into columns of the data frame, and then took the count values from each one and put it in there. Okay. So now I have a nice wide data frame for the female transmission category. Same thing for male, exactly the same code. Same thing. And now I want to put them all together. And I'm going to do that by just doing bind rows, because they all have the same. That's work. Yeah, it's going to work, but it's going to give you needs. Okay. So you see, I did bind rows. Some of, like, for example, the child data frame has only has only one group. Only one column, only like so many columns. So where their columns matched, we get numbers. So here's the injection drug use, right? So that's in both the male and female. But some of these don't have values, so there are going to be NAs where they didn't match up. Does that make sense? They don't have exactly the same number of columns in each group. So where they match up names, they matched up and found them, and where they didn't, NAs got that in.
Yeah. What's the difference between using bind and merge? Like, what's the benefit of using bind?
That's a good question. I don't think I've used merge.
I think it's a base R.
Yeah. If I've used merge, it would have been a very long time ago. Yeah. I'm sorry, I can't. That's a Richard.
Yeah, well, so merge is very much like a join statement, right? Like left join, right join. So if you have like a shared variable between the two, like two data frames, like ID or something like that, like TID or something. That's when maybe you want to use merge or like left join, right join.
Oh, okay, yeah.
Join.
Could you join? He goes this way.
Right, right. And it's like if say one data frame is like 100 rows, the other data frame is only like 50 rows, right? Like depending on how you merge, you may get different dimensions as the output. So we don't.
Yeah.
I don't think we went.
No, I don't think we did. And again, because we did some joins, right? The joins, yeah, joins kind of just match up this way. Will they also add? I don't know.
It depends. It
Yeah,
depends
just
if you're doing like a left or right or full. Or inner,
yeah, but generally, right. Generally, when we do this, when we're combining data frames these days, we're using bind rows, bind columns, or joins, depending on what your intention is. And we probably could have done a join in here too, but bind rows works too.
Okay. Sure. Yeah. So join. Sorry. Join within tidyverse like auto detects the columns that it's going to join on. So if there's like columns with the same names, it just
You just figure
joins
it out.
them and says, hey, merge, you have to specify what they are.
Okay.
Which is not, I have mixed feelings about that. On the one hand, I'm lazy.
Yeah.
I don't want to type any more than I have to type. On the other hand, using assumed behavior can be dangerous. Especially like when they come like along and change the assumed behavior of a function, you know, for whatever reason, you know, oh, this seemed like it didn't work out so well, so we want to do something else. And then your code breaks and yeah. So it's probably better to be more explicit. Like if you're doing a join and even though it has the same name, you specify them.
Yeah.
Yeah. We don't though, because we're like
Exactly.
all right, so now we've got this thing. And wait, do I not remove the NAs yet? All right. There. Okay, so we've got this transmission data frame, we've got a bunch of NAs in there. We do want to make it a little bit more tidy. So these two transmission. So we're going to take we're going to do a pivot longer. So instead of having this wide data frame, we're going to take the transmission and put the take the values and put them in count. So this is the type of transmission for each category. So I've got male and all of their types of transmission, and then you'll see the same thing somewhere down the line for female. Get to it. There we go. Okay. All right. And now, so now we've got a nice tidy data frame, but we've got some NAs. So we're going to get rid of some NAs. Yeah, and I'm repeating all the code because I actually haven't saved this at all. I don't know if you noticed in the piece cells above, I just ran the pipe, but I didn't save it into a new data frame. So now we're going to save it. We're repeating the pivot longer thing, typing that to NA omit on count, and saving that in the transmission data frame. And now we can see it. Okay. So it's nice and clean, no NAs in there anymore. And it's tidy. And now we can do some analyses. So one thing we can do is we can summarize. So we can group by, so remember, I don't remember the summarize and group by and summarize paradigm. I use this all the time. It's very, very useful. So I'm taking the data frame, type it, say, I want to group by transmission, and then summarize the number of deaths is the sum on the count variable. And it gives me this really nice data. Questions so far? No, so far, all right. Okay, I'm gonna do some plots. And did you ever facet grid and stuff? You did, okay, well, it's worth seeing it again. So here we're gonna plot, remember ggplot has this aesthetic thing, so you have to give it a mapping. So aesthetic is how do I map my data to the geometry of the plot. So X is going to be transmission, Y is going to be the count variable, and still equal transmission. So this isn't actually giving me more. Is it? Yeah, this isn't actually giving me more information. It's just going to give me a nice legend and pretty colors on the map, on the plot. So, and then we're going to do this thing called facet grid, which is going to split everything up. So it's going to give me one plot for each year and category pair. So I get a nice grid. I'm going to do a bar plot. Stat equals identity. This is what you do when you want a bar plot and you already have count data, because you can also do bar plots when you don't have count data, but then you have to use a different stat. And I don't know how much you remember, but stats and geoms go together. So so if you have so the stat for geom bar would be count. If you don't have count data, you do, then you just say identity, because basically saying don't do anything, just use this as count. And this is just a theme, and this is to get rid of the X text and the ticks. So let's run this. Ta-da! See, we don't have values on the X-axis. So what we did, yeah. So we don't have values. That was what our text element equals blank for these things. So we don't have ticks and we don't have values on the X-axis. But that's okay, because our X value is transmission, and we made this nice legend here. Oh, I guess I can't talk to the whole thing on the screen. There we go, that's better. Okay. So child, male, female, male, and then the years, and then all of the different transmission categories. But I hope you can appreciate how nice this comes out. You just all you have to do is get the data in the right format, and then the ggplot does everything for you, and so do the dplyr. Oh, and yes, yeah, we don't talk about cable, KABL in here. I don't think it's on the notes, but so we show you how to make nice plots. But there's also this command called cable that makes really nice tables. And this is in tidyverse. Oh man. I'm actually a pure mathematician, so I like math. But this means it's in that table. Cable is a function in tidyverse. I think actually I don't know which. It's just part of cable. Cable is a package.
Knitr. KNITR.
Oh, it's in knitr.
Yes.
So it's in knitr, part of tidyverse. But then there's also something called cable extra.
EXT.
EXTR.
Yeah.
and I think the E is capitalized. And that gives you even more stuff. And if you knit your Markdown notebook to HTML, you can put like slide bars and you can do hovers on your tables, and so it's really, really nice. So it's a little note that, you know, when you're looking to do more, that's a really great package. Okay, so let's do another plot. This time we're going to do a facet wrap on transmission, and we're going to smooth. We're going to do just basically a scatter plot, but with a smoothing. So you can ask to have, so you can do your scatter plot, and you can ask ggplot to just draw a nice smoothing curve that looks that kind of reflects the data. There are many ways to smooth data, get smooth curves. The default in GeoSmooth is something called LOS, which is a kind of a running linear regression that does over windows, and it's just smooth that way. Oh, and I saved it in G. And then this is just to show you that. So here's another R chunk in the notebook that can, so in your R chunk, you can specify how big you want what the dimensions you want your figure to be. And suppressed warnings is because that's when you know, because sometimes ggplot will ignore NAs in your data. We don't have any here, but if you did have NAs, it will print a warning for every NA it drops, and it's a little bit annoying because hopefully if you have NAs, you know that they're there. If you don't know that they're there, then you haven't spend enough time looking at your data. Okay. So what does it look like? Here's what it looks like. No way I will get this whole thing on one screen, but so here's the transmission categories that we broke down, and the smoothed counts. And I don't really like too happy with this figure. So you guys like, first of all, you can't see the whole thing on one screen, so it's kind of a pain. But also like that we have, and it looks like the count axis label is in a weird place. So I don't know why that happened. But anyway, this was to show you that you can also use facet wrap, which creates facet wrap creates a plot for each of your ordinal variables. So you've got transmission as an ordinal variable here. So for each value, it will create an individual plot for it. All right. Any questions? I'm going to give you some to do for yourselves. So you can just start working and questions.
Yeah, just Well,
I just want to point out that the last one is really hard, so if you don't want to like, you just want me to do the demonstration. Think about it, but some of the things are not, we haven't even covered. Are most of you still working? It's hard to tell because for all I know, you're already working on your. Conversation. How's everybody doing? Anybody done? Kind of. Like, I don't want to touch question 4. Is that okay? Should I give you a few more minutes or should I go on? Go on? Okay, cool. All right. So, yeah. So the first couple hopefully weren't too bad. The last one really does need you to kind of dig in a little bit. So if you didn't quite get there, don't worry about it. Reading in the Excel file, hopefully that was pretty straightforward because it's a nice simple Excel file. So you just read it. How many columns are there? Where are the columns? That's the dimension command, right? So that's right there. And then the third one is the number of agents with HIV growing over time. Just so you have to do a few things here. First of all, you want to pull out the part of the data frame that relates to race and ethnicity, because it's the same structure as the other one, but the data sets, it's got the categories and the groups. So the first thing that I did actually, I said, well, okay, let me actually have a look. And hopefully you did this too, at what categories they have, what groups they have in race. Because if you look carefully, you'll see there are 2 Asian categories. Why? I don't know. I don't know what the asterisk means. There's probably somewhere where this data was pulled from, there's probably some sort of data dictionary that explains that, but I don't know why it is. But that means that you actually want to get fancy and use that string detect command that I showed you before. Because, or you could also say, you could also do an or statement. You could say group filter group equal Asian star and group or group equal Asian star. So you don't have to use string detect, but it is a quick way to do it. So here is just pulling out, oops, sorry, pulling out all of the rows that are the race category and with the group Asian, saving them in this data frame DX Asian. And then for the bar plot, all you have to do is the aesthetics are year for X, and then the count is Y. And I did fill equal year so I can get the colors, and then geom bar. And again, just like we did before with the stat identity. And it looks like that. Any questions so far? Anything weird about this? Anybody notice? Besides the fact that I neglected to put in a title, floppy and lazy. But anything look weird about the legend and the coloring?
Gradient.
Yeah, why is it a gradient? Anybody know? Because year in the data frame, which is my x-axis, here's my tibble. So the really nice thing about tibbles, when you print them out, you can see what type R thinks each of your variables are. It thinks that year is a double, or in other words, a decimal number, so a continuous variable. Year is actually a factor. So I can do that. I can change this by doing a little mutate here. So before I say this, I'm going to say mutate year equal, I think I can do it this way. I mean, if it doesn't work, I'm going to have to try a different syntax, but I think this works. And let's just see if that works. Yeah, okay. Okay, so when you see this, this is so fun. This is, I think this came up when I was TAing for Francis. Says year not found. And you note that I didn't put year in quotes. This is where you run into, like, especially, so factor is a base R command. So especially the base R commands, they always want you to, they want the column names in quotes. Unless you're doing a $sign to address them. So there's also like, but anyway, if you come up with something like this and you're like, no, year is there, it's definitely there. Try quotes. Okay, I'm gonna do it differently. I'm gonna do as underscore factor. Yes, okay. Huh. But I'm not finding it. Oh, did anybody see my error? I just found it. I somehow wiped out my pipe. Okay, and so now you see year says factor FCTR like that. So, and I'm gonna rerun the entire chunk now. Oops. Oh, did it? Do I have to specify the levels? That's very strange. Isn't it fun to watch the teacher make mistakes? Ew. I think it might be that let me do it this way. Oh, maybe, oh. Okay. So if you use factor, you need to have quotes around here. If you use as underscore factor, because it is a new declare tidyverse, I know it's not declare, it was forecast, but it's a new tidyverse thing. You don't have quotes, and if you put the quotes in there, you get weird things. So here is, so if I took the quotes away, and now it works. I hope that's instructive, because it's the kind of thing where you just like, oh, just take the quotes off, put the quotes in. All right, so now it's a factor, and you see what you expect, like the coloring as you expect it to be. Any questions? Okay, this is the last part. So make a horizontal bar chart of the total number of people with HIV over all years for each ethnic group. All right, there should be a single Asian group. The bar chart should, so that's like a lot, right? Okay, so here's the first thing. So the first thing we can do is we can use the mutate command to change the group category so that there's only one Asian category. And it's a little bit, this is why I said this is advanced. The string replace command will replace, we'll take this column group, and when it finds this Asian dot star, that means anything that has the Asian and then star after, which would mean both the Asian star and the Asian star star. This is something called, it's something called a regular expression in strings. If you ever have to really deal with a lot of string work, you should learn regular expression. Completely beyond the scope of what we do in this workshop. But what happens is I hate them. They're weird and cryptic, and every time I use them, I learn them, and then I forget about them completely. And then the next time I have to use them, you learn the whole thing, and I just sit there going, hate this. But anyway, the dot means anything that this thing and then follow. So the point is that it will cover both Asian star and Asian star star, and then it replaces it with the next string, which is this one. So we do that, we take out only the race ethnicity category, grouped by the different groups within there. And because now we've got fixes, so there's only one. And then we can summarize, oh, is this deaths? Wait a minute. People living with AIDS. Yeah, so sorry. So this is a typo. This should be, I don't know. Let's just say EWH. Okay, so there we have a nice table that has the counts that we want. And then if we do H again, so this is the same code again. And if we want to arrange in descending order, we put this just give this to descend here. So arrange. So I'm going to arrange it in descending order. Making group a factor. So just what I did before, except this time it's using the factor command. Oh, I wonder if it's going to even run with the. I guess we'll find out. Yeah. That's up. Okay, that's how the factor thing works. I don't know why. And then so we changed group to a factor, and then so that it would break it up easily on in the bar plot, and then the very last thing we did was forward flip. So we asked for a horizontal forward plot. The forward flip just takes the axes and moves them. And there it is. I want to see this as. And I'll post the solutions on the repository so you have them. Okay. So that's the end of this part of this particular example, that data mining part. I suggest we maybe take a break and then come back, unless you want to keep going, because I know you kind of y'all want to take a break, like 10 minutes. Okay, 10 minutes. So come back at 25 of, and we will do the really fun sequencing example. And by fun, I mean frustrating. A little frustrating.
Could you scroll up to just that last bit of again? We started on I know.
we have to put it into programs. But to get people to continue with it gives you this, it's not, what it was called. Yeah, that's what there's like something else. But anyway, little like that, it's actually that. And then it like shows you, and sometimes there's a thing that because it makes sense. You're, I'm sure it's just within okay, but the point is is the rate majority of it not sure what they're not it should be in order to do that you know the original thing the experts that's why I know you sequence the funders to do no, actually it's not continue it. Yeah. And then we download that. Yeah. And then we align it. Yeah, just to make sure. Right. Again,
So technically that was correct, because there was one.
Violet and yes.
No,
I think it's not. And I'm not saying that it's security. But you're right.
wait, why does it matter?
The only one that we was that alarm was.
Those are the only ones we use. Did you hear Nolan? Yeah. He was trying to be general.
Was that the one?
Did he go?
But I will when they. But yeah. But he was for a while. And like,
To ask someone.
I didn't.
I did like.
I have a black. No. I know, I didn't cheat. There's the other aspect.
this was just by the actual.
So. I don't. I usually, I stack like a couple in the hood and then like I get like 3 or 4 all of them. And then that's not very helpful. That way there's not like a bunch of things that go over it. But it's annoying and if you want that you're right. Yeah. No, you're okay. Right. Well, what did I do? I need some more. I have yeah, like then like this. No. So. So the earliest. But tomorrow's. Oh, yeah. So. They're really. Reaching for my screen, just hurt my lower back. Oh, the 3D thing, yeah. Right there on our, I mean, that's. Yeah. Someone warned me. Yeah, right, so you were different. For the next one, I didn't practice, no, I got it, James, like, about family. Absolutely, are they, if you want to get I heard that like, no, I want to say. Going up to there. I've never done like before. There's so many like little things that I like. Oh my God. You know that you covered. Yeah. Yeah, you like rather. I know because I checked that's the boarding. So much fun. No, I so
I mentioned before, and Richard on these things and reminded me that to bring your own data. If you have stuff with PHI, I'm probably already know this, but just I forgot to think about that. PHI, you can't bring it to me. You can't put it on the DCC. Does anybody have a PHI data set that they really kind of want to help with, or no? Oh, good. So it's not a problem. Don't forget about that. I just recently started a project, not recently, well, about a year ago, with the EHR data. And oh my God, what a mess. I mean, it's a mess, it's de-identified, but then we have to re-identify some people, and then I had to get onto Citrix and get like, and pay lots of money. Yeah, it's like, I think it was like $400 just to have like a Pace workstation, and then it was more money to get the code. Like, this is great. But anyway, it's important. I think it's super important that we're careful with that kind of stuff, but it's also it can be such a bottleneck. Anyways, my rant on that. Let's go. Did I move the whole thing? Yeah. I lost my whole. Okay, we're back. Sometimes it just kind of disconnects from the server. Okay, so go back up a directory, and then we were in practical, and now we're going to go into the sequence pipe. Okay. And the RMD file is the HIV alignment database.rmd. So here we're going to work with this is LANL, this is Los Alamos National Laboratory. They have, do any of you work with genomics in HIV? No, wow, okay. Maybe I should find an example. Anyway, they have sequences, and this particular experiment is it's one patient, but they have written here. It's longitudinal data set of viral epitopes and T cell measures for one patient. And there are cell spreadsheets corresponding to clinical data. So they have the CD4 and CD8 counts, plus viral load. And then they have counts of epitopes that are strongly selected, weakly selected, etc. So it's drug resistance. I'm not really sure actually I remember that properly. They also have FASTA files, which you could use to get alignment accounts, but we're not going to do that here. We're not going to be working with that. If you are continuing the workshop, and the one that's in the spring, I'm pretty sure they're doing the assay workshop. Definitely going to include some kind of sequencing and alignment sort of stuff. Okay. So first, just do the setup chunk. So this, by the way, is a setup chunk. And I don't know if anyone described these to you previously, but in the R notebook, you can not just have an R chunk, but you can have like specific ones that are for specific things. So a setup chunk is one that sets everything for the entire notebook. And so if you've worked in R, base R before, you can set your working directory. This is the equivalent of that for root. So in a notebook, you don't do that just because of the way that they're executed. Set working directory isn't the thing that you want to do. What you want to do is in a setup chunk, put root directory. I have it in here, although to tell you the truth, I actually use absolute path names throughout the notebook, so I don't really have to have it in here at all. But if you want to use a working directory, that's how to do it in the notebook. Okay, so I'm going to give you a block of code. I'm not going to go through the details of it. The block of code figures out a path, like sets up a path with a lot of pasting commands and downloads a file from the website. I have, so this is basically for your reference later. Okay, I have first this code that's all commented out in this chunk. This is stuff that I just got from a help forum, probably Stack Exchange. You know, but and that's we do that a lot. When you want to do something hard, you don't know how to do it. Once you've learned the sort of the basics, then you're like, oh, how do I get a file, you know, download from the web in R? And that's what I did, and I got this chunk. And then I had to change a bunch of things from my specific example, and those are here. So for example, the URL is, this is the URL where the file is that contains the data that I want. So don't worry about all the stuff that's done in here, because it's actually already here. So you can execute it, but the files are actually already on the repo. So should be able to. But there's one last thing. Okay, so here's basically I got all the files and then file names, and then I used map, which we haven't talked about, right? No one talked about map. What this whole command is doing, and this is a little bit beyond the scope, but I'm going to say about it. I'm going to tell you about it anyway. What this command is doing is it's saying, okay, I've got a list of file names, right? That's the thing called files, and it's saying, I want you to read these file names over. So it's iterating over the list for me. So that's what map takes a list and it does something, whatever function you give it. So the function I gave it is read Excel. So it's going to apply that function to every element of the list. This is an advanced form of, we call it vectorization. And if you're going to really get into R at some point, you should learn about vectorization, because that's how you do things. And actually, it doesn't work so well with the map functions, but there's also something called lapply, which does exactly, it's the older version of the map, but you can do something called multi-core lapply. So if you want to do really, really easy parallel programming, you can just use MCLapply and it will take your function, your list, and for each element of the list, it'll assign it to a core. So I use that when I'm, say, doing like really big data fitting things that take a lot of time. So each one might take minutes. So, and if I want to do 40 of them and I'm running on the cluster, I can do them all at once. So that's just another aside. And then what it does, and so in the end, it stores each of these in a list. So it's reading each file using read Excel, and it's putting it into an element, each one, a data frame in a list. So now I have df list as a list of data frames. And when I printed it out, like by typing it on a line by itself, which we print things in R, I get things that look like this. So this is nice. So this is the first one, the first data frame, and this is clinical data. So days post first onset of acute symptoms, CD4 counts, CD8 counts. This is great. This is exactly the kind of like format I want to see my data, and it's got one header line, it's got columns, it's all tidy, great. This one, yeah, I hate that these are so, they're very, very wide. Yeah, this one is bad. So I'm supposed to have like magnitude of CTL responses. There's one main header, and then there's like other gobbledygook here, right? And this is actually the epitope names, but and then so you can see that something weird here, right? So now I'm going to do what I should have done to begin with, which is go look at the files. I think unfortunately we have to download them now. I'm just copy pasting here. So this is at the sequence database, and if you download the zip file, so this is the zip file, and this is what it contains. So I said I told you this clinical data, there's FAFSA files, So this is all the stuff that I read all the Excel files in my map command. But if I want to look things, strongly selected episodes. Yeah, so this is kind of a mess, right? I mean, it's not, I mean, if you're just gonna look at it in an Excel file, right, it looks fine. But the problem is, when I use read Excel, it says, okay, the first line is my header. And then the second line is data. And so it just said, oh, okay, everybody has the same, we don't have header lines except for the first one, right? It said, okay, the first column gets this header magnitude CPL. Then it named everybody else. And then it said everybody's character because there's stuff in here that's not numeric, right? And it stuffed like the header line into the data. And we don't want that, right? Okay, that's one example. So this is one format. Also, we have another like weird thing here is that days post symptom onset covers like these 3 header lines here. And yeah, so this whole column has a different format from the rest of the file. Okay. There's another one. Look at variant one. Yes, okay. And so this format, again, we have this DPS thing here. We have original and variant strings as headers, right? And then we have the epitopes underneath. So this again, like when Read Excel sees this, it doesn't know what to do with it. It's not gonna process it properly. So, but the good thing is, when there are things we can do, and we're going to show you things that we can do to get the information that we need, because in some cases, like we need the information that's in the header, and in other cases we don't, and yeah. That's what they look like. Our studio. Okay. So now you know why Gazette didn't get itself. One of the reasons. And you can say, well, why don't I just copy and paste? Well, we don't do that because we want everything to be reproducible. So, and we need some of that information that's in the headers, right? We just don't want to throw them away. We need to format them somehow. All right. So, and I just described here, I'm not going to read all that text here to you. I just told you. So there are basically 3 types of formatting. There's a really nice clinical data set. Then there's the one that just has the extra header line and the weird thing with DPS. And then there's the one that has a variant original complication to it. So we're going to figure out how to do this. So first we're going to start with the strongly selected episode file. That's not the one with the variant in it. It's the other one. All right. So, and I'm going to do things that are wrong on purpose. Okay. All right. So the first thing I'm going to do is okay, let's get rid of the first header line, right? It doesn't tell me anything other than I might want that information for something, but I don't want it for now. I mean, it's just giving me the units I think that they use for the accounts. So that's what skip does. Skip equal 1 here. N max equal 2. Is supposed to just read 2 lines. So let's see what happens when we do this. Okay, so we got rid of the first line. That's great. But we still have 2 header lines. So we have the first header line. So now we have the column names for the epitopes, which is nice. But these the second header line is really supposed to go with the first header line, right? It's the same sort of information. And again, it thinks everything's a character because that. Okay, so we can tell Read Excel to ignore. I'm sorry. Oh, I'm sorry. The first thing that we're going to address, I forgot, is that we have this header here, the one that spans several rows. This one header. Okay, so we can do that. We can tell Read Excel to ignore the first column, and then we can go back and get it later. Okay. So we use the range option there. So we skip the first header line, read to, and the range is B through I. So, and I think you do have to give it the Excel names for the columns in Read Excel, which is a little bit of a pain, but especially if you're like me and you don't use Excel a lot and you're like, wait a minute, what are the names? A, what? So, okay. So, that's strange, right? I've got this, remember, this was the first line and I told it to skip. Right? I said, skip the first one. Well, what happened was the range option overrode the and max option and the skip. So it messed us up. So now what we have to do is take the just manually extract the first two rows. Oh, sorry. Oh, okay. I forgot to change this. Oh, I'm sorry. You guys are gonna have the same problem. All right. So if you follow me, just like, actually, want to do, let's just erase the entire thing. Erase the whole thing. All right. Start with a tilde. Then hit the tab key. What? Oh, sorry, tilde slash. Then tilde slash, and it should show you what directories you have in there. You might only have the one. I have a bunch of stuff because I use this container for a lot. So that's that workshop. Then hit tab again and put data science in. Tab again. Examples. Tab again. Sequence. Tab again. Files. One more tab. And we want to go all the way down to strongly selected activities. We'll take that. I use tab completion all the time. I strongly encourage you to do it because it will prevent you from making typos that will fix your code. And then sometimes R does weird things like setting things to null. And like I had once a typo where it was like I had transposed the letters on a variable name, and it was in like 150 lines of code. I couldn't find it anywhere. And so yeah. Trauma. Okay. All right, so now we'll execute this and hopefully it will work. Okay. All right, so wait a minute, this is weird. It's not working the way it's supposed to. It's not skipping. Oh no, this is fine, sorry. These are the column is sorry. So we have these headers. I don't care about the headers. What I wanted was the columns, the column names, these two rows. So this is fine. So we're going to do, the next thing we're going to do is we're going to use this string C to concatenate the two strings together across the thing, across those columns. And so that's what this looks like. Okay. So now we have like the first header, and then the second header, and then the first header, second header. Clear to everyone and what happened there. Okay. Now we can extract the data into it. So we're gonna ignore. So we pulled out our header lines, right? That we want to make our headers, and then we're going to ignore the headers in the Excel file and say, okay, just give me the data in the rows. So, determine where it goes here. Okay. Oh, I'm so sorry. I just, looks like I got the next one. I somehow missed these two cells in changing the file names. So basically the same thing. as before, if you don't want to do the tab thing, just come up and like copy this file name from here and paste it down into the next one. Sorry about that, that's annoying. What's kind of funny is I said to myself, I was like, oh, that only happened in a few places. I thought there were more, and there were. Okay. All right, so here what we're going to do is, so it's the same file name. We're skipping 3. So I'm going to skip all the header lines, and I'm going to define the column names. This is all in the read Excel function. Define the column names to be the vector, the string vector DPS plus those column strong that I also selected out. Okay. Everybody got that so far? It's gonna, okay. So I'm setting the headers and reading the data in all at once. Okay. This is much more what we want. Okay. So I've got date post symptom onset. I've got each of the epitopes and the counts for those epitopes. Right? Okay. There is one little issue though. See, read Excel, just like all the other like read R families of functions, they assign types. And the way that they do that actually really probably didn't really talk about that, but they read a bunch of lines from the file, like 1,000, I think is the default, and then they look to see, do I only see numbers? Oh, I only see numbers, then it's going to be a number. But if there's like something in there that's weird, like a character, or it's something that's supposed to be an NA but doesn't, like sometimes like quote NA in R is different from NA. NA is a keyword. If quotes around NA, now it's a string. So things like that. So something in here made it think that DPS was a character. You can see the CHR here. And I don't want that, because if I want to make plots and things like that, it's just, I really want my my time variable to be numeric. Okay, so the way that we do that, so I'm going to do all of this the same thing, and we are going to this time. So I'm repeating the whole read command, and I'm adding a new. So one way we could do it was we could use the mutate command and use as integer, and I think we've shown that in other examples. So I can mutate the column in the data frame that way. Another thing, but with read Excel, I can actually tell them what the column type should be. So here I'm saying the first column, I want you to consider that to be numeric. And then this rep guess length of column name strong. Column name strong, remember, is we have DPS and then column name strong with everybody else. So what this code is saying is the first one is numeric, and then repeat guess that many number of times. So I'm giving it a vector that is the length of the number of columns in the data frame. And I'm saying first column is numeric, guess everybody else. Because it was guessing everybody else, fine. We just the first one. There we go. So like I said, there's always more than one way to do things. And one way is to get the data frame in and then mutate the column there. But you can also just, tell the read command what you know the column instead of letting it guess. Okay. Yeah, that's right. And then so in the Excel file, actually, the last, let's see these NAs in here? They happen because I want to pull the Excel file back up for you. If I can, I'm just going to do the download thing again. Not that way. No, actually, I don't do that. I want to show you what they did in here. Strongly selected epitope. See that at the bottom of the file? They said, oh yeah, they posted onset DPS means date post onset of acute symptom. And that's, that was what messed us up with the GPS being parsed as being numeric, right? Because it was, but it's in the first column right here, right? So it's kind of a pain. So don't do that in your Excel file. At least not when you give them the last message.
I have a question.
Yeah.
Sometimes in your variable, you add comments like, for example, the GPS, instead of putting that line at the very end, we sometimes put it like a comment in. Write the same thing that
In Excel?
they did, yes. Yes. Instead of writing this explanation about what GPS means, sometimes people add a comment in the variable.
Yeah,
That comment interfere with whatever.
I haven't seen that. I would not be surprised if Read Excel can handle that. Probably better than just putting the little thing at the bottom, because Excel is flagging that as a comment. Better yet, and Richard, chime in if you think that I'm wrong, but I would say the better thing to do is just to have a data dictionary that explains every column in your data set, right?
Yeah,
And that's either in a separate tab or a separate file.
yeah.
Yeah.
So like a comment, like in a comment on a cell in Excel.
Yeah,
Yeah, I don't know. I don't know if it read that in.
this is Read Excel.
Yeah, read it.
Yeah,
Does it?
it's not from Read R, it's a different package.
Yeah, but
Okay.
I don't think it reads any comments.
Okay.
I don't think. I know it. Or if it does, it's a pain.
Yeah, but the best way to do it really is to have a data dictionary. Yeah. Right. Okay, and so, oh yeah, Read Excel even told me about it. Look at that. Said I was expecting a numeric, but I got this. So it didn't automatically put the, oh no, because I told it I want numeric. That's why I gave you the warning. Didn't give me the warning the first time. It just said, okay, there's a character. But the second time it said, you want numeric, and I've got something in here that's not numeric, and I put NAs in there. So it's good to get those warnings are nice, so that in case you didn't know that that's what was there. All right, and so I want to get rid of those NA lines, so I use filter not, so that the exclamation point means not, is .NA of GPS. So you never ask in R, you don't say, is this thing equal to NA? Remember what I just said before, NA in quotes is a string. And without quotes, it's a keyword, but you don't say equal to NA, you use this function is.NA. And so get rid of those. And now I have a pretty clean data set. And oh, like I made a plot here that I forgot about. Okay. Yep. Okay. And so these are counts for each of the different more. I thought there were more. I guess that's it. Okay, so I'm just using oh, I guess I shouldn't like kind of go through this a little bit more carefully. So let's just look at what strongly selected looks like now. Okay. So if I want to do to make those plots that I showed you before, yeah. If I want to get this output, I actually have to make this wide data frame into a long data frame, because I'm gonna want to say I'm gonna want to have a category variable that says which epitope it is, and then I can pass it back over that. Okay. So I am going to take this format and just make it longer. Get rid of days post symptom onset as a column. It should be, sorry. To just to get the longer, the long format, I'm taking the days out. The names are epitope. And so basically what, oh, sorry. I'm getting tired, can you tell? Pivot longer, I'm telling you which columns I want to apply this to. The columns I want to apply it to is everything except DPS. So instead of listing out all the columns I want to deal with, I just say minus DPS, right? Everybody else is an epitope. I'm going to store the names in epitope. So I'm going to take the column names that I'm pivoting, put them in epitope, and the values I'm going to put in a column called CTL. And then, so these aren't GCAPs actually, sorry, these are the cytotoxic cells. And then we're plotting on x-axis days post symptom onset, y-axis is going to be the CTL values and coloring by epitope, and then plus geom points that tells it to make a scatter plot. And that, sorry. And then I'm faceting by, I'm doing facet wrap over epitope, and that's giving me all the different plot values that I have there. Somehow thought there were more epitopes not in the file, but I guess I'm wrong. Okay. So, any questions? All right, great. Now you get to do it. And I have the wrong file name here again. So I want you to get the variant end epitopes from the directory. And again, it's going to be, let's go all the way back. You've got tilde slash tab key, the workshop tab, data science tab, sample tab, my voice, sequence tab, files tab. Yeah, master directory tabs. Okay. And I wanted, excuse me, variant add end epitope. Oh, I'm getting ahead of myself. That's the next part of my lecture. God. What I wanted you to do actually here is you do the exercise with the files that have the same structure. So you don't have to do it for everyone. Just find another file that has the same structure and do it. And try not to copy and paste too much, right? Look at, it's probably better for you if you look at what I've done and then retype it rather than just sort of cutting off. It's better, like if your brain will retain it more because if you just add copy and paste it. Okay. Go ahead. Are most of you still working? We got a lot. Is anyone done? All right, so you'd like more time to keep your work? I just want to make sure I'm not There's only about 20 minutes left, and I was going to go through this one last example. Those of you, yeah, all of you actually, what do you find most challenging about this exercise? Or is it just tedious and thinking? Anyone?
I would say the most difficult part was, to grasp was extracting and creating the new, with the characters for the first and second rows of the file to then merge them.
Oh, the
together.
concatenation
Yeah.
was hard to
Yeah,
understand.
just hard to conceptualize.
Okay.
Yeah.
Yeah, I guess because it's one of those things that, yeah, it's a vectorized thing, right? And so yeah, I can see that. I'm careful about explaining that next time. Anything else? Will you ever make another Excel file with multiple variables? My work is done. So I'm just gonna go on. I just want to show you this last one. And then, next time. Well, actually, before we even do that, because I seem to be losing people a little bit. So next time we're gonna do bring your own data. Those of you who are going to bring who have data in mind, can you tell me a little bit about what it is? Yeah,
Like single cell data?
single cell data. Okay, but you got the counts. Secret. Okay, very good.
We both have data from mice that it's just their weight over subsequent weeks. So it seems relatively simple compared to this.
Okay, cool. Mice infected with, I'm not familiar with.
Tuberculosis infected mice.
Oh, okay, okay, because, okay, I actually always think that everyone's doing HIV, but this is
Yeah.
TB. Okay. All right. Anyone else? Any data? Yeah.
Mine is also not HIV. It's a grad school project that I was doing. It's HDL function in Japanese. People across and I also have their BMI, I have lipid values, I have
So
demographic
HDL
data.
being, sorry,
Cholesterol. Yeah,
okay.
cholesterol.
Mhm.
Function of cholesterol.
And you're looking at, right?
I'm looking at to see how the oxidation is affected by different types of medications and diet. And then I did a previous analysis with Americans, so I want to compare my previous analysis to the Japanese because theirs is way better than ours.
That sounds fun. Cool. All right, and the rest of you, what kind of examples would you like to work on? Do you have anything in particular that you'd like to have more practice on, or? Yeah,
Is there a way to upload fast Q files? Because I'm working on sequencing data with microRNA, but we actually have fast Q files, so I don't know how to upload that to R.
I mean, there isn't, to my knowledge, there
Yeah.
isn't a way to read FSQ into R. I don't think there's an
Okay.
R library for it. I know, and it's been a while, I do teach in the sequencing workshop that we do over the summer. And basically all of the, like the reading of the past cues and the alignments and the counts, that's all done outside of R. It's done, so the use star, that's what we were using for the alignment program. And then I can't remember what we were using for counts. I think it was a Python script. And then, yes, a star is a C program that's compiled, you run that on the command line, and then there's a Python script for counts. And then finally you go in and you do DEC in R. Yeah. Anyone else? Because I'll try to come up with some fun examples on my own, but if you guys have anything particular, I'm happy to tailor to what it is that you'd like to see.
Oh, I have some like microbiome data where I'm trying to correlate like a biomarker to microbiome.
Okay.
So I guess it's like a counts table,
Uh-huh.
but I want to look at like whether certain microbes are like co-enriched like at the same time point, if that makes sense. Like I want to see if they correlate with a biomarker, but then within that time
But at
point,
the time point,
like are there, like which ones are correlating and like in what direction? Like if there are groups of them that like increase together or decrease together. Okay, that makes sense. I don't know.
bring it
Okay.
and we'll figure it out. It's usually easier for me when I'm sitting down and somebody has the data and I can look at what it is exactly, like what they look at and then how to put together. All right, well, let's do this one very last thing, last few minutes. It's very similar. It's just that here now we have this added complication that they have the original epitope and then they have variant epitopes. And so now we've got like 4 header lines. Again, the days post symptom is here like on its own. And then they have the main header line, which just basically says the units that we're using. And then original and variant. The one thing that helps us is that for the original guys, they have this I guess this is, it has I don't know what actually this description is. It's none of you do genomics, so you don't know either. But anyway, the original has this extra string on it that identifies the epitope, as opposed to the variants, which only have the sequence. Okay, so these are the amino acid sequences I believe here. And so the way that I'm going to know whether a column is representing an original or a variant is not by looking at here, because the read Excel program can't see that this cell is spread out over just many columns. All it's going to do is it's going to take the first one and it's going to call it variant, and everybody else is going to give it a made up name. Okay, so I'm going to use the fact that this string is present here and not present here to create sort of a column vector that tells me whether we've got a variant or an original. So yeah, so this is just reading in. The first one is just reading in the data to get at we're skipping the first 3. And so now we're just getting this thing where we have the thing where I can tell where I've got, wait, sorry, I might be ahead of myself. Nope, okay. Good. Here it is. Okay, so this is how we're going to figure out whether it's an original or variant. So first thing I'm going to do is just read in the column names, read in skipping the first 3, and then getting that last header line. Show you what it looks like. Oh, that's cool. All right. So here's what I want to look at. I've got original and variant, and then a bunch of NAs, and then the different the amino acid sequence, and then for the original, I've got the epitope tag. And so this next line of code, this if else command that I know we've introduced you to before, but just to remind you, if else takes a logical, so expression. So this is going to be evaluated to either true or false. And if it's true, it returns the first thing. If it's false, it returns the second thing. So what we end up with is this looks at all the column names of, here, I'll actually show you what it is that we're looking at here. The first row. So this is the first row of that of the thing that I read in, and it looks like this. So if it's NA, then it's a variant, right? So filling in the blanks essentially, replacing NAs with variant, and then if it's not NA, it's original. See what it looks like. So now I have a vector that tells me that says that flags original variant, original variant. Okay. And now this is the thing that you said was confusing. We're going to concatenate for the two rows for original where we have the amino acid sequence and the epitope name. So again, if it's not, we have these if else, and so if it's not NA, because some of them are, then the very variant ones are NA, then we're going to concatenate the second and third ones. And that's what we get. So now we can see that we have concatenated the amino acids with the amino acid sequence with the epitope names. And now bring it together with the thing we just made up. So now we have a tag original or variant with the sequence and the the sequence and the epitope tag. And now we just put it all together. So I'll read it all in. And then essentially do exactly what I just said. And of course, fix this, get rid of the NAs that are in the date close into the onset like we did before. So it's basically exactly the same thing that I did. Oh, right, it's the warning. Right, that warning. Exactly the same thing that I did with the other file, except dealing with this other like original variant thing from the other header line. So if you want to look at that, you should look at it. If you don't want to do this exercise, then I don't think it's actually necessary. But it's just to show you like how you deal with something like that in a reproducible way. Richard says he's doing something very similar to that right now. Okay. So you can either stay and do the exercise, stay and ask me questions, or you are free to go.